Good morning to us students. Today is Wednesday, March 18, 2020. We're gonna start with a new chapter, Chapter 4, Biological Identity and Genotype. In order to start with Activity 1, Phenotypes and Proteins, please open your books to page 76. The objective of this activity is to examine the origin of some phenotypic abnormalities or diseases in order to prove the relation between proteins and phenotypes. As a recap to what we took in Chapter 3, the genes or the DNA is maintained by replication. Then it transforms into RNA by transcription process, which transforms to amino acid sequences by translation process. Then the obtained proteins are divided or classified into either cellular or functional proteins. These proteins are going to be considered as traits. The traits can either be antigens like HLA or the biological markers of cells, and blood groups, which specifies the blood group of an individual. Or they can be pigments like skin, hair, or eye color pigments. Then these traits are going to be expressed in the individual, which is going to be known as phenotypes. It is the set of all the hidden or visible traits or characteristics that are expressed in an individual. Now, when talking about abnormalities in proteins, for example, in functional proteins, there are certain enzymes that are considered functional proteins and are responsible for the production of melanin pigment. What is melanin? Melanin is a pigment that is responsible for the hair and skin color. Now, its absence leads to albinism, which is a hereditary disease. Two examples of an albino boy and girl are shown in the next pictures. Now, melanin is synthesized in the organism from tyrosine, which is an amino acid precursor. The melanin pigment is produced due to the action of the functional proteins, which are the enzymes 1 and 2 shown in the next document. As you can see, the two amino acids, phenylalanine and tyrosine, that are obtained from our diet are actually going to lead to the production of melanin. Phenylalanine is supposed to be transformed into tyrosine amino acid by the action of enzyme 1, and tyrosine by itself is supposed to transform into the melanin pigment by the action of enzyme 2. So any abnormality at the level of the gene that is responsible for coding for enzymes 1 and 2 can lead to an abnormality in the transformation of the amino acids into melanin pigment, which can lead to its absence and therefore shows the abnormal phenotype, albino phenotype. Application for functional proteins. Given document 1 showing the production of melanin pigment and showing the consequences of having a normal melanin pigment and in the case of the absence of this pigment. Read document 1 really well before starting with the questions. Question number 1. Pick out from document 1 the involved protein and the abnormal phenotype due to its absence. Answer. Any of enzymes 1 or 2 are the involved proteins, and their abnormality leads to the absence of melanin, which leads to albinism. Question number 2. Formulate two hypotheses at the origin of this abnormality. So we should think, what would be the reason behind having an abnormal or an absence of melanin pigment? One of the reasons could be that enzyme 1, which is the functional protein that has a huge role in producing melanin, could be non-functional. So hypothesis 1 would be enzyme 1 deficiency. Another very good hypothesis can be set, which is enzyme 2 deficiency, as also enzyme 2 has a huge role in producing melanin pigment. Question number 3. We inject the albino person with tyrosine. 3.1. If the albino recovered his pigment, then the problem is due to what? So we should think, if we had an absence of melanin pigment and as a recovery process tyrosine was injected, it is as if we added a molecule that compensated the loss of enzyme 1, if enzyme 1 was actually lost. And this would lead to an appearance of melanin pigment. Therefore, the problem would be in the absence of enzyme 1. 3.2. If the albino couldn't recover his pigment, then the problem is due to what? In the case of the absence of melanin pigment, although tyrosine was injected, this would indicate that the problem is after tyrosine and not before tyrosine. After tyrosine, enzyme 2 is in action that is able to transform tyrosine into the melanin pigment. And therefore, by the production of melanin pigment, enzyme 2 is considered to be functional. However, as given in 3.2, albino couldn't recover his pigment. This means that the cause of this absence is due to enzyme 2. Structural proteins. One very famous example of structural proteins is the dystrophin protein. Dystrophin is a protein that contributes to building the membrane of muscle fibers. The lack of dystrophin protein from the muscles leads to a disease characterized by muscular degeneration, and this disease is called Duchenne muscular myopathy. This disease mainly attacks boys, and it is a hereditary disease. Another example of structural proteins is the beta-globin protein. A normal hemoglobin, known as HbA, which carries oxygen normally in the blood, is a protein that consists of 2 normal alpha globins and 2 normal beta globins. An abnormal hemoglobin, known as HbS, is characterized by having an abnormal beta globin protein, which differs by a single amino acid from the normal beta globin protein. This abnormality leads to a hereditary disease which is called sickle cell anemia and is characterized by an abnormally shaped red blood cell, as shown in the neighboring document. Application for structural proteins. Given in document 1, the consequences of the presence of dystrophin and its absence. Muscle cells produce dystrophin protein, which interferes in the structural constitution of muscle fibers. However, in the absence of dystrophin proteins, weak muscle fibers are produced, which leads to atrophy or degeneration of muscles, leading to muscle paralysis. The muscle paralysis is called Duchenne muscular dystrophy, DMD myopathy. Question number 1. Pick out the involved protein and the abnormal phenotype resulting from it. Answer. The involved protein is dystrophin protein, and its absence involves a muscular paralysis. Question number 2. Formulate a hypothesis at the origin of this abnormality. A very reasonable hypothesis would be mutation in the DNA, which codes for the synthesis of dystrophin protein, leads to DMD. Kindly copy the previous and the following notes along with this homework on your copybook, and please solve this homework for next time. Now, in order to start with Activity 3, the genotype, please open your books to page 80. What is a genotype? A genotype is the whole set of genes for the combination of the different alleles carried by an individual. As shown in the left side of the neighboring document, one chromosome can carry a whole bunch of genes. Most living beings are diploid and carry two copies, by which we call alleles of the same gene. Each of these alleles is present on a homologous chromosome. As shown in the right side of the neighboring document, a homologous pair, one having a paternal origin and one having a maternal origin. Each chromosome carries one type of allele. Allele 1 is carried by the paternal origin, in this case chromosome, and allele 2 is carried by the maternal origin chromosome. The phenotype is the expression either of one or two alleles when dominant, or both when codominant or of an intermediary dominance. The allele that is expressed is the dominant allele, and the allele that is masked is the recessive one. In this example, we're considering a pair of chromosome number 9. The first chromosome carries allele 1 and the other one carries allele 2, and we are interested here in the gene responsible for the blood group trait. The genotype can either be homozygote, in other words pure, which means having identical alleles, or heterozygote or hybrid, which means having two different alleles. Taking into consideration that in case of different alleles, just the strong dominant allele will be expressed and appears at birth, we symbolize it by a capital letter, while the other weak recessive allele will just be masked, which is symbolized by a small letter. For the ABO system, which codes for blood groups, three alleles are present, A, B, and O. A is dominant over O, B is dominant over O, and A and B are codominant with respect to each other. A small application is shown in the next table.